Concept 44.4, the nephron is organized for stepwise processing of blood filtrate. And we will go through the steps in varying levels of detail depending on where we are at within the nephron. Here I've just provided you with a microscopy image of the nephron, so you can see glomerulus here, and then you can see all of these tubules, right? So this still kind of adheres to the description of the excretory system being a network of tubules. So we'll work our way through the nephron. And as we think about the components of the nephron, you want to consider what things are found within filtrate, what things should not be present in filtrate by the time we get to the end of the process, what mechanisms are utilized in order to achieve filtrate formation and filtrate modification, but still just on the level that I'll discuss them within the voice thread. So this is really more for some of my students that have already had anatomy and physiology. This is important. The level of detail is not the same for this class. Now, how we start off is through the Bowman's capsule and the glomerulus. Okay, so in this illustration, we can see a bunch of things going on. So I will walk through what's being shown here, and you're responsible for all of the vocabulary that you see here. So the filtrate produced in the Bowman's capsule, which is essentially within this glomerular space, is going to consist of salts and glucose, amino acids, vitamins, waste, other small molecules. Now, with that list of things that you hear, what that is also going to indicate to you is that the filtrate contains some things that sound beneficial, such as glucose, which we know can be used to drive metabolic processes, or vitamins. So consider the events that are taking place at this location and what is then essential as a result of that. Okay, so the glomerular capsule is going to be continuous with the renal tubule here, and the very first portion of the renal tubule is the proximal tubule. When it comes to the glomerulus, which you can see here is a kind of a large knot, it's made up of a bunch of fenestrated capillaries. And to be fenestrated simply means that the capillaries are pretty porous. And the porous nature of the capillaries allows for large amounts of solute-rich but protein-free fluid to exit out of the blood and enter into the glomerular space. So this fluid is the filtrate. Okay, so things are being forced out of these fenestrated capillaries. So these are these outward arrows, and they're entering into this glomerular space, which is surrounded by this glomerular capsule, just to mention what is shown there. Now, when it comes to the blood flow, we can see that blood is entering through into the glomerulus to the afferent arteriole, and it's exiting out, E for exit, through the efferent arteriole. There is, just because it's really conspicuous in the illustration, there is a difference with respect to diameter between these arterioles, and that does have an impact on the rate of filtration that takes place. But that's not a topic for principles of biology. So I'm just acknowledging that there is a difference between the arterioles that does have an impact on blood flow, but the concept and explanation of that we won't discuss here. Now, when it comes to glomerular filtration, it's a passive process. So how we're able to generate the filtrate that's found within the space is going to be through the pressure of blood flowing through the capillaries. And that is going to force fluids and the solutes through the membrane of the capillary. So there's no direct consumption of metabolic energy for this process. Now, if it's a passive process and we're strictly, focusing on or generating our filtrate based on that force, then you can see here that we don't have a means for regulation at this time. So we're just forcing things out, and our level of regulation is essentially based strictly on size. And when it comes to that size, there's essentially a filtration membrane, and there's several steps to it. And your book just goes through one of the layers of filtration selectivity, but there are actually three. So this is the podocytes that you see here. That's the smallest one. But there are capillary pores and there are collagen fibers in addition to this. And all of those collectively help to prevent the exiting out of formed elements. They will help retain glycoproteins as a result of charge. They'll keep albumin inside as well. So there is a level of filtration based on size, but we can say, okay, well, glucose is something that I need, so retain the glucose as well. The process can't do that. Okay, so we can restrict certain things, but we can't restrict everything. And as a result, the filtrate that exits out into this glomerular space absolutely needs to be modified before it exits the body. Otherwise, there would be a loss of things that are essential for us. So at this point, we're generating filtrate. We have some level of selection, but it's based solely on size. We can't hold on to the things that we need at this point, so the filtrate is going to contain both the good and the bad. But from here, we'll then work our way through the renal tubule where there will be some level of modification that begins to take place. Here we have net filtration pressure. And when it comes to the ability to form that filtrate that is taking place at the glomerulus, there are several factors that are involved in that, and I want to address all of them. So just to recap, you have your afferent arteriole bringing blood into these fenestrated capillaries, and the force of that blood flow allows for components of blood, restrictions based on size, to exit out into the capsular space. And then that filtrate can eventually make its way through the renal tubule, starting with the proximal tubule. Now, when it comes to this process, it is more than just the hydrostatic pressure that you see here. There are going to be forces that promote or favor filtration, so meaning they favor the movement of filtrate out into the capsular space. And then there will be processes that oppose it. So they are going to result in filtrate moving out of the capsular space back into the fenestrated capillaries rather than through the renal tubule. So first we have the glomerular blood hydrostatic pressure, and that's determined mostly by systemic blood pressure. So that will favor filtration. So it's going to favor the movement of substances into the capsular space. Now, what we'll see here, just quantitatively compared to the others, is that this hydrostatic pressure is going to be higher than the other factors, okay, at least in this particular example. So the hydrostatic pressure seen here is higher than the capsular hydrostatic pressure and the blood colloid osmotic pressure. And that's important when it comes to our calculations. Second is the capsular hydrostatic pressure. So in capsular hydrostatic pressure, this force is going to oppose filtration because what this is essentially associated with is you have a bunch of blood that's rushing in and it's going to rush out and start to fill this space. But there's only so much fluid that can move into the renal tubule, so there will be some buildup within the capsular space. And as a result of that pressure, some fluid or some filtrate will move back into the capillaries. So as a result of the capsular space rapidly filling up with filtrate, some of that fluid will go back into the capillaries, and that's going to be an opposing force of filtration because that movement backwards is not contributing towards the filtrate that exits out into the renal tubule. The third is blood colloid osmotic pressure. And for this osmotic pressure, this is created mostly by albumin. And this is going to oppose filtration by pulling water back into the glomerular capillaries. And that's because if we take this and do a little bit of drawing here, if I let the letter A be albumin, then we have a bunch of that within these blood vessels, okay? And because there was restrictions that were based on size, it's going to remain in the blood vessels. So what we would then expect is, okay, water's going to say, well, there's a ton of the albumin within the blood vessels, and there's none out here. So water is going to want to flow back in to that vessel as a result of that difference in solute concentration. So you have protein within the blood vessel that's at a much higher concentration there than it is within the capsular space. So in response, water is going to flow back into the capillaries. Okay, so that is osmosis. So that is why it's referred to as osmotic pressure. So all three of these factors contribute to the net filtration pressure. And because the blood hydrostatic pressure is higher than both of the others combined, we still have a value that favors filtration. And so essentially, anything that favors filtration will be a positive integer. Something that opposes filtration will be a negative integer. So we can see that the capsular hydrostatic pressure here is negative. The colloid osmotic pressure here is negative, right? So these are opposing the process of filtration, but your hydrostatic pressure is positive. And when we do the calculation for this, which is going to be the hydrostatic pressure minus the capsular hydrostatic pressure minus the osmotic pressure, that's going to let us know, whether or not the process of filtration can occur. You always want this integer to be positive. And the net filtration pressure is one of the main controllable factors of the glomerular filtration rate. And all of that means is the amount of filtrate that the kidney can form in a certain amount of time. So specifically within one minute. So within this illustration, you want to know, what are these three things doing in this process? When are they, which ones are favoring, which ones are opposing? How do you calculate net filtration pressure? What does it mean to be a positive integer, to be a negative integer? And now that you know how these three things work, how they're defined, think about possible ways that the process can be changed and how might that impact the values that we see for these different components? How might that impact the net filtration pressure overall? What type of diseases or disorders could impact the net filtration pressure in a negative way? Those are just some things off the top of my head that you'd want to consider based on the information from this slide. The renal tubule is going to be the duct that leads away from the glomerular capsule, and it's divided into distinct regions. Now, some will collectively incorporate this lower portion. Some will break it up into two. So you might see some books that say there are 3 regions versus those that say there are 4. Now, I'm really going to place more of the emphasis on the transition from one cell type to another as we work our way through this tubule. So when it comes to these regions, there is going to be the proximal tubule. So the proximal tubule is the longest. It has very prominent microvilli, so those projections or extensions, and this supports a high amount of absorption. It also promotes secretion at this site. And the cells that we'll find here will be cuboidal epithelial cells, and there will be some large mitochondria there. So when we're looking at this first segment, it's the longest. It's got a ton of coiling that's here. So some books will refer to it as the convoluted tubule or the PCT. Here, your book is kind of consistently referring to things as just proximal tubule. So that's how I will refer to things as we work our way through our discussion. After we continue or work our way through the proximal tubule, we'll head into the nephron loop. And the nephron loop is made up of a descending limb and an ascending limb. And when it comes to the nephron loop, which is sometimes referred to as the loop of Henle, there will be thin segments and there will be thick segments. The thin segment is simple squamous, and that's what makes it thinner. And there's going to be low metabolic activity here, but there will be permeability to water. And the statement will be a lot more relevant when we talk about osmolarity and regulation of urine concentration. The thick segment is going to be composed of cuboidal epithelium, and there is going to be a high amount of mitochondria here. The cells will be thicker. There will be a high utilization of active transport. So there will be a lot of metabolic activity that occurs in this region of the nephron loop. So after we go from proximal tubule through the nephron loop, descending, and then working our way up again, we will head to the distal tubule. The distal tubule will also be cuboidal, and it will be confined to the renal cortex of the kidney. And this will mark the end of the nephron, which is why we have it grayed out over here, because this would have been the next portion in which we head to. When we use the terms proximal tubule and distal tubule, these names are going to be based on their relationship to the renal corpuscle. So just to give you a frame of reference. So if you're thinking about where do I go to next, proximal will be closest to the renal corpuscle, distal will be furthest away from it. As filtrate makes its way through the nephron, which is the grayed out portion in this illustration, it will eventually head towards the collecting system. And the collecting system is composed of different types of collecting ducts based on their location. So some found within the cortex, some found within the medulla here. And so the collecting duct receives fluid from the distal tubule of several nephrons as it passes back into the medulla, and then tons of collecting ducts will converge towards the medullary pyramid. There's a pyramid shape to the medulla area. If you go back and take a look at the kidney frontal section, you can get a sense of that organization there, because obviously you can't see that in this one. But they will converge near that pyramid, and that's where they'll form a larger papillary duct. And so they're about, between 25 and 30 papillary ducts, and they'll end in pores. And these pores will be found in an area that's not indicated by your textbook. It's called a calyx. You don't really need to know it for your lecture exam. Other than that, that's where we would be heading to next here. And so ultimately, urine is going to drain from the pores into the calyces. And then, from there, we will make our way towards the renal pelvis. So this is kind of a more specific statement to my earlier one of renal pelvis and other associated regions. Okay, so what you want to take from this is the collecting system components you can find in the cortex, you can find in the medulla. I'm not really acknowledging the histology of the different regions. I'm not going to test you on that. We will use this particular figure to go over the reabsorptive capabilities of the renal tubule and the collecting duct. So the ability of these different portions within the kidney to reclaim things that had initially entered the filtrate through glomerular filtration. And when it comes to the renal tubule, the entire thing is involved in reabsorptive processes, but the proximal tubule is by far the most active location of reabsorption that occurs. And so what we find at the proximal tubule is that pretty much all of the glucose that should not leave the body can get reclaimed in this area. You would expect for large portions of amino acids and other organic solutes to also be reclaimed here. A large amount of water is reclaimed here. There are a ton of ions that are reclaimed here as well. And within the process of reabsorption, there's an active component to that. So there is going to be some active transport as shown by the red arrow. So there is a requirement of ATP, whether that ATP utilization is direct or indirect, that needs to be present in order for these events to occur. But there will also be some passive tubular reabsorption, and that will take place through diffusion or facilitated diffusion or the process of osmosis. When you go past the proximal tubule, things change pretty significantly when it comes to permeability and events that take place. So collectively, in just thinking about the nephron loop, the main things that we focus on are water and salt. And the behavior that takes place here is really important for the concentration aspect of the kidneys and how that relates to urine production. Now, when looking at the nephron loop, water reabsorption is not coupled to solute reabsorption. So water can leave the descending limb, so heading down here, of the nephron loop, but water cannot leave the ascending side. So there's inhibition of water movement on the ascending side as compared to the descending side. Now, that's because there are essentially no aquaporins or very few aquaporins, but essentially no water movement is taking place on that side. as a reminder, aquaporins are an example of a channel protein. This was discussed in Bio 100 more from the point of what type of amino acids would you expect to see lining the interior channel versus the side that's closer to the phospholipid bilayers. Now we're thinking about the aquaporins really in the role that they play in terms of allowing water to move. But remember, aquaporins are channels, and so that movement is going to be passive. So you have the ability for water to move out on the descending side. So you can see that it's a passive process, but there's no ability for water to move out on the ascending side. And these differences in permeability are important for our ability to adjust the level of concentration that we see in urine. For solutes, what we see is the opposite, which is that very much no solute reabsorption occurs on the descending side, but solutes are reabsorbed both passively as well as actively in the ascending limb. Now, when we look at the proximal tubule, we see a lot going on with respect to diversity of what is going into the tubule versus what's going out of the tubule. And what we can see when we're at the nephron loop is that it kind of just focuses then on the role of water and the role of sodium chloride as far as reabsorption goes. By the time we get to the distal tubule, this is now an opportunity to fine-tune the process of reabsorption. And this is really where we determine what that urine is going to be composed of. The point that's here, as well as the collecting duct, are times in which hormones can come in to modify or regulate the status of the filtrate. Now, most of the water and solutes are already reabsorbed by the time we arrive at the distal tubule, so it's really only a small amount, and that is why we say fine-tuning. So just, in thinking about this for preparing your studying, I don't necessarily love this particular Campbell image, but a lot of the images that I utilize for other courses has too much information for your class. So I would say for the content that's gone over here, the actual text, just the reading material, is a lot more beneficial than just simply looking at this, because there are a lot of things that are missing here. I don't think it's necessarily clear what these arrows are showing in terms of in or out. But I would just say, in thinking about what you want to take from the image, you want to think about, what are my different areas? Notice that I've combined the nephron loop as one distinct thing, but what's going on at each distinct area? Where is the bulk of reabsorption taking place? Where is the gradient being established? What's happening at the distal tubule? Where are my last opportunities for filtrate modification? Those are questions that you want to provide answers to as you're going through it. But just read through the text that goes through these numbered sections, because I think that's a little bit more clear than what this visual is providing.